It's nice to see so many familiar and newly seen faces. So also it's been a while since I gave a Zoom talk. So this is yeah, it's good to go back to the Zoom roots, I guess. Even though it also is nice to be in person again. So I will talk a bit about some of the work that we've been conducting during the past 4 to 5 years, which really tries to look at how abiotic stress, and we will get to what exactly we mean by that, affects organism. And I do this currently in the capacity of environmental toxicology. That's the department where I'm housed here at Uppsala University in Sweden. So this, of course, by definition, then in this case also includes chemical perturbations, but this is not what I will be talking about today. But I think this community surely knows this, but I wanted to reiterate that environments are complex and multidimensional, and this is something that many fields struggle with, particularly by knowing this, also environmental toxicology. And I think it is well encapsulated by the dynamic processes, for example, shown here by this very well known cubic millimeter of seawater, where many of these biological interactions unfold over very, very small spatial scales. So for one, there's a tremendous amount of biological heterogeneity that is not just pertinent to species-species differences, which I think we all can recognize and be sure about that these matter, but also even within the same species that phenotypic heterogeneity plays a huge role. So even if the molecular signatures, in a way, the genotype, the blueprint by which cells are built is exactly the same, the expression of certain genes and other components ultimately leads to stochasticity in phenotypes, which are can be adapted or less adapted to specific conditions. And of course, many perhaps of you also look at taxes, so kind of discontinuous kind of systems in which, for example, if a bacteria finds this nutrient plume that is exuded by a copepod, might be benefiting from that, and if it doesn't, it does not. And I think that directly relates to an underappreciated kind of segment of this, which is really life history. So I think through our the way that we have used molecular tools to provide snapshot measurements of certain environments, I think what we are not really taking into account is, for example, how life history effects might play a role here. So very concretely, say 2 phytoplankton cells, of which one is exposed to a stressor before, might be more sensitive to a subsequent stressor. And so this complex biological variability is ongoing on the background of dynamic abiotic environments, which fluctuate dynamically over a range of timescales ranging from milliseconds to decades, and include, for example, temperature, oxygen, pH, light, and really mediate some of the biological variability described earlier. And so many fields try this, but in my particular case, what we are trying in my group is to actually understand how pollution now, when it enters this particular network, affects one specific type of organism, and how this relates to perhaps even ecosystem impact. So you can see this is a daunting task. It's complicated. There's many interactions here that are that we perhaps even have no understanding of yet. So what we're doing in my lab is basically trying to take a reductionist approach to select a few of these abiotic stressors, which kind of are encapsulated in these abiotic environments, and combine it with assessments of biological heterogeneity in marine microorganisms. And we chose this group specifically because they play a dominant and huge role in biogeochemical cycles, for example, carbon fixation and sequestration to the deep ocean. So any finding that we might have here might have ecosystem-wide implications. And to do this, we really try to develop integrated approaches and methodologies, both to recreate, but also to quantify abiotic environments. And so the 2 cornerstone technologies that we use are microfabrication in a broader sense and microsensing. So the bottom line is we recreate some of these abiotic environments in the lab. Here, a selected few that we are currently working on or I have worked on in the past. We're actively engaging, for example, in the recreation of oxygen regimes that are really exact replicas of natural environments. But today I will be mostly talking about temperature, because it's really truly a mediator of metabolism. In these ectothermic organisms that are directly exposed to certain temperatures, the metabolic rates might change immediately. And so typically we look at these as singular exposures, so kind of taking one of these stressors, testing it very thoroughly, but the long-term ambition, and we are also engaging in this now, is to create multi-stressor regimes where many of these compounds, these abiotic stressors, are combined in a realistic way. And we also developed technologies then to sense some of these abiotic environments and also to assess the metabolic effects by, for example, developing single-cell respiration technologies. So today I will be mostly talking about basically the use of some exemplary projects where we used microfluidics to test the effect of abiotic stress, in this case only temperature, on certain microorganismal responses. And then I will deviate slightly and introduce you to a technology that is relatively recent, that we published in 2022, which we call Sense PIV, which are chemically sensing particles that allow us to sense oxygen inflow across complex biological architectures. And you can see a video sequence here where we inject an anoxic solution into an oxic solution and visualize that by basic camera systems. So coming to abiotic stress then, and how we can use microfluidics to recreate them and ultimately look at and kind of chart microorganismal responses. So how do we actually do this? This was work that I still did during my postdoc together with Roman. Basically, this was a microfluidic device that we developed, which was called phenotype, which really has three principal design features that kind of permeate much of my work in the follow-up. So one is we typically create microwells, so here depicted by number 1, into which we can place individual microorganisms. In this case, you can see here one well containing one of these microalgae that we are trying to investigate. So the photosynthetic unicells that are of interest, which I will describe in a minute. And so we have many of these wells, about 30,000, and we can immobilize, of course, many of these cells. Sometimes we have duplets, sometimes we have triplets, but we also have a significant amount of individual cells that remain stationary. And basically, the advantage of that is that we can observe the same cell over time. And then we can expose these cells that are immobilized in this central channel to certain environmental perturbations. In this case, we can use, here depicted in number 2, this side channel into which we can inject, for example, a silver-laced epoxy polymer. And by applying then an electrical stimulus, we can basically heat locally. So you can see this depicted down here. On this side, we have this reservoir acts as a heating source. And on the opposing side, we have a heating sink, which is basically just the same channel geometry through which we flow regular water. So we can basically generate and dissipate heat, and we can create a linear gradient of temperature across these immobilized cells located in the central channel here. We can combine this or do it individually by exposing these cells also to chemistry. In this case, we use this Christmas tree gradient generator to create a gradient of chemistry going from a high to a low concentration, which is superimposed on top of these arrays of individual cells that we can then observe. And of course, this microfluidic device, because it's so small, must be coupled to a microfluidic readout. And I'm not going to go into the great detail of how exactly the technology works, but basically we're using imaging chlorophyll fluorometry, so PAM fluorometry for those of you who are familiar with it, to observe the photosynthetic efficiency of individual cells. So these are 4 microalgae, as you can see them here, immobilized in these microwells, and they show a different color, and that relates directly to their photophysiological capacity. So now I talked a bit about kind of microalgae and that we're using them in a microfluidic context and to test abiotic stressors. So what are these microalgae? And I think they warrant a brief introduction in 1 or 2 slides here. So one of our model systems in my lab are Symbionacea cells. For those of you who are not familiar with them, these are coral symbionts. So coral reefs being these biological megastructures that you can actually see from outer space. But if you zoom in onto the microscale, what you can see is that corals are basically a symbiosis between a photosynthetic unicell, these are the small red dots in this picture on the left here, that sit inside the animal host, which are kind of these finger-like protrusions, which are the polyps themselves. And so these are also called the engine of the reef because they are photosynthetically active, they harvest light and give much of the energy that are harvested by them back to the coral for survival. So this is what it is. It's called Symbiodinium, the coral symbiont. These are dinoflagellates with a beautiful golden brown color. And the nice thing about them is that you can actually cultivate them in the lab. So you can take them out from corals, you can bring them into the laboratory, and you can grow them actually to do experimental manipulations with. And so this is important into the context of climate change or basically coral bleaching in general. And this describes the fact that a healthy coral contains some of these symbiotic cells, these photosynthetic unit cells, but then due to exposure of a multitude of different factors, including and mostly dominantly temperature, this symbiosis becomes disrupted, leaving the coral basically in a bleached state where it does not contain corals anymore. Symbiotic cells. And so understanding ultimately how these individual cells of Symbiotidaceae react to external perturbation in abiotic environments, such as temperature, irradiance, pH, has potential implications for our understanding of coral reef survival under these upcoming stressor regimes. And just to give you an insight on what that actually looks like when you bleach a coral, this is a time-lapse photography sequence where we exposed these corals to a very, very strong Q of temperature. But what you can basically see is that these corals react quite dramatically. And of course, this is an accelerated phenomena here, condensed maybe 3 to 4 months total time into maybe 9 hours in this video sequence. So our one fundamental question that we are trying to answer is ultimately how individuals of Symbiotidaceae cells from different species respond to temperature stress. This is one of the research projects that we have ongoing. And so we started off by probing this question by actually this first device that I showed you, where we basically used it to expose these cells to dynamic temperature stress. So in this case, what I'm showing you here is now cells that sit inside the central channel, which we are exposing to 5 heating and cooling cycles at a different temperature magnitude. And so this is an average because it represents basically the temperature at the central part of the channel. So some cells will experience a higher temperature the closer you are to the heating source, and some will experience a lower temperature when you are to the dissipating element. And on the y-axis, we have FVO of M, which is photosynthetic efficiency of cells, 1 being 100%, so being perfect in conducting photosynthesis, and 0 being photophysiologically inactive. And we can increase now the magnitude of the perturbation while we repeat this dynamic process. And because this is an imaging readout, what we can actually do is we can visualize these processes immediately. So on the top here now we have the heating source, at the bottom we have the heating sink, and this is the legend for these individual cells ranging from 0 photosynthetic efficiency to 100% photosynthetic efficiency. And so what you can gain an appreciation for is that these processes are dynamic. There's a heterogeneity in these populations. You can see that some cells react more strongly to the external temperature perturbation than others. So, for example, keep an eye on this corner here. There's probably 1 or 2 cells that are actually surviving these rather extreme temperature perturbations better than their sister cells. This can be quantified as averages, so basically across this channel geometry, closer or further away to the heating element. And what we can see is that these populations respond quite dynamically to the external cue, but also that they have the possibility to recover from them. So I think what we learned a lot from doing these experiments, but one thing that we also learned was that kind of mechanistically and technically, we were a bit limited in terms of the stressor applications that we can actually supply to these cells. So in my lab, then we teamed up with a local collaborator, Maria Tenye, and we basically together developed a jewel-based heating system. As you can see here, this is a 2-component glass system, which contains a cooling channel below and integrated heaters and temperature sensors with an optical window in between. And this is linked then to a PCB board, which is computer controlled. This is a cross-section of this device now also sandwiched together with a microfluidic chip that sits on top. And this is where the cells are basically placed. So similar, we again do microwell immobilization, and then through a thin layer of immersion oil, we can bring these into close contact with each other. And through careful calibration, we can ensure that the heating output here is the one actually reaching the microfluidic device. And so this allows us then to really expose cells to much more user-defined temperatures than what was previously possible using the simpler chip geometry. So just to give you a couple of examples of some of the temperature profiles that can be executed and also supplied to cells, this is a classic stepwise increasing temperature regime where we go from the nominal cultivation temperature of some of these cells, 22 degrees, by increasing every 15 minutes by 1 degree Celsius. This is typically used by us to determine thresholds of cells. We can supply more artificial temperature regimes such as rapid ramps and cooling cycles to in a way understand the recovery dynamics of some of these cells. And perhaps what I'm most excited about is actually to recreate complex natural cycles. So this is actual sensor data that was captured in situ by a sensor buoy on a tropical coral reef that we downloaded and then recreated in situ over a 24-hour cycle. So this is basically a diel cycle of temperature that is experienced by corals in their actual environment, and we can recreate that in the lab. And of course, we can also supply them as gradients, similar to the previous device, where we go from a low to a very high temperature. And due to the large size of this device, we can also basically assess the effect of these temperature perturbations on many thousands of cells. So my postdoc Lindong then used this particular Joule-based heating system to ultimately probe the temperature sensitivity of different Symbiodinaceae species. She applied this stepwise increase in terms of temperature to these cells and measured the reduction in their photosynthetic efficiency. So what you see here in this heat map are now 5 different symbiont species exposed to increasing temperatures, and what we measure is basically the reduction in photosynthetic efficiencies compared to the control. So basically, we're taking 22% as 100%, and then subsequently the reduction is then related to the colorimetric change as displayed here. And what we can see is, and not so surprising, I think, but it was a good confirmation, is that these species differ from one another, and that some are less temperature sensitive. Stars here denote the first time that there's a significant difference between the actual control and the reduction in photosynthetic efficiency between these populations, and that some are much, much more temperature sensitive. This can be and I think where it became exciting for us then is applying really toxicological principles to this, where we now calculate the thermal dose, the accumulative thermal dose that these cells experience, and measure the amount of photosynthetically inactive cells as a result of that. And that allows us to calculate something called the effective distress dose, where basically 50% of the cells are succumbing to the effect of that particular temperature stressor, basically a reflection of the heat map that you see here, but in a way, a really easy readout. And so one of the things that we're trying to pursue now, can we actually go into the field, extract some of these Symbiodinaceae species, put them into our devices, and perform such an assessment in a way, not in the field, but close to the field. But I think the real power of this technology, of course, comes into play when you can then look at individual cells. So this is one of these heat map plots here over time, where you can basically see that each individual dot represents one cell, that the photosynthetic efficiency in the beginning is quite normally distributed. This is a normal distribution fit, but then as we are advancing and progressing through the temperature stress regime, that distribution kind of falls apart, really telling us that ultimately that that subpopulations exist in these particular populations, that some of which are better at withstanding temperature stress and some are not. This is a pattern that we repeatedly saw, that basically once these thresholds, so for example, around 34 degrees Celsius are reached, this is typically where heterogeneity plays a huge role, meaning really that phenotypic heterogeneity in a way goes undiscovered in these particular species. If standard temperatures are applied, but once high and kind of surpassing the threshold temperatures are applied, the phenotypic heterogeneity widens, it becomes larger. This is recreated here. So you can see on top the 5 different species of Symbiodinaceae that we tested against their photosynthetic efficiency at the bottom. Again, each dot representing a single cell under increasing temperatures. And so we can see and gain an appreciation for that distributions typically wide in some of these species. And I think what we're excited about now is actually probing some of these specific phenotypes that, for example, convey high thermal resiliency. So, for example, marked down here. And this is because due to the arrayed nature of these devices, what we can actually do is we can selectively retrieve individual cells from that particular array. In this case, we use laser capture microdissection to create a small microbubble in a way to catapult the cell into the fluid stream, allowing us then subsequently to retrieve them. What we're excited about is now actually linking the phenotype that we observe to the actual genotypes, or the actual molecular signatures that make up that particular phenotype. So I hope this gave you a bit of an insight into how we can recreate and supply abiotic stressors, in this case temperature, to cells in order to understand their responses, and also really see and perhaps delineate how environmental variability and phenotypic heterogeneity interplay here. So now we're going to go into a different part of my talk where we will talk about sense-PIV, this technology that actually allows us to sense oxygen across complex biological architectures. And so perhaps for this audience, this is a bit more the ballpark that you're normally working in. Basically, these are, so measuring flow is probably something that you might be doing using particle velocimetry. Essentially, by providing tracer particles at a high enough density, one can basically measure and recreate and measure 2D vector flow fields that really describe flow dynamics in different systems. So for example, on the left side, a starfish larvae, or on the right side here, a more complex topography by coral polyps. Now, other people then also describe oxygen and the distribution thereof. So pioneering work by Helle Plog, for example, where she used a microsensor to basically probe profiles of individual measurements using electrochemical or amperometric sensors and measure locally the oxygen concentration around a sinking particle. So really fantastic work. The problem with some of these technologies is that it takes a long time to basically perform these types of measurements. This is in part circumvented by planar optodes. I will get into what these exactly are in a minute, but these basically to allow us to measure, and for example, Ronnie Glut has used them to measure here the oxygen distribution on top of a sediment in blue, the bottom of a sediment in black, and then also how bioirrigation by burrowing worms ultimately might actually lead to the contribution of oxygen into these sediments. And then more recent work by other groups in Denmark, for example, where they used nanoparticles in this bioirrigation setup also to track and trace, in a way, fluid streams with oxygen. And I think the latter paper was really an inspiration for us in a way to ask the question, can we combine measurements of fluid flow with that of oxygen at a local scale? And so this is where sense-PIV was born, which basically some fundamental idea, can we perform simultaneous measurements of flow and oxygen in a manner as described down here? So basically, can we create particles that once they cross an oxic or go from an oxic into an anoxic barrier will yield a colorimetric change that we can pick up and measure quantitatively? And so sense-PIV basically now combines oxygen measurements with particle velocimetry and allows us to estimate diffusers as well as advective oxygen fluxes at small scale and small time scales. And this is really work that's been driven by also these two people, so Klaus Koren being the sensor chemist at Aarhus University, and Søren, a collaborator at the Max Planck Institute. So coming back to these optodes, how do we actually sense oxygen inflow? So here's a model particle, and basically it constitutes of a two-layer shell. So one in the middle, this is the core. This green part is basically an oxygen insensitive dye. So this dye will not react to any external oxygen. And in red, we have a shell here, which is basically this particular chemical, so PTTFPP, which is actually oxygen sensitive. And this allows us to actually do ratiometric measurements between an oxygen insensitive, so the reference, and an oxygen sensitive dye. How do we do this? So basically, these sensor chemistries absorb both light. Typically, we apply UV light. That leads to an excited state and the release of that excess energy through luminescence. So this is in the absence of oxygen. Basically, here the luminescence intensity and the reference intensity is equivalent, one being green, the other one being red. And the nice part of this, we can actually capture this with a simple RGB camera because we can split the channels up afterwards. Now, in the presence of oxygen, what happens is that more and more oxygen molecules actually interfere with the de-excitation and lead to luminescence quenching, but only for that of PTTFPP. So basically, only the sensor chemistry in this case is affected. This is in part shown down here, where on the y-axis we have intensity and the corresponding luminescence intensity as a function of increasing oxygen concentration. So typically we have the highest density at the lowest oxygen concentration. Now, Klaus has made these sensor particles through two different methods. I'm not going to go into the details, but I want to highlight here that staining is basically just the coating of existing particles, and precipitation is a different method that actually allows them to produce liters and liters of these particles, which can be used for more demanding applications where many of these particles are used. And here's just a calibration graph where we basically used both these ratiometric precipitated particles as well as the luminescent stained particles and measured under different oxygen concentration their luminescence intensity ratio at 0% oxygen divided by that at a given oxygen concentration. And this actually allowed us to do simple 2-point calibrations for these sensor particles and apply them for 2 different setups. And so the one that we started with was actually a microfluidic system because it provided full control over kind of the oxygen concentration and a very stable system in a way to determine and test whether sense-PIV actually works. So in this case, we have a microfluidic channel with an oxygen source and an oxygen sink, resulting in a linear gradient of oxygen between these flowing particles. And so the resulting data looks like this. Across this channel position, we can see these dotted lines are the oxygen concentration in % air saturation at different time intervals, so 50 seconds, 1 minute and 20 seconds, and so on, and the corresponding model fit using COMSOL. And at the same time, we can also measure the velocity profile here, the parabolic regime due to wall effects, very much matching the simulation by the computer. So this gave us confidence that we could apply these sense-PIV particles to other more complex biological architectures. And we started off by actually applying this to corals. So in this case, we put a small coral fragment into a flow chamber, used a light sheet laser to ultimately capture, in the presence of some of these sense-PIV particles, the RGB signal by a simple RGB camera. This is what that looks like in somewhat real life. Here you can see the sense-PIV particles as they flow over the coral surface. There's a microsensor placed there as well to verify the oxygen concentration locally. And you can see the diffusive boundary layers down here and the ciliary movement that actually moves particles in that diffusive boundary layer. Now, through the combination of both, what we can now do is actually, in this case here, create a combined map of the flow along the coral surface. So one being generated by particle tracking velocimetry and the other one by particle image velocimetry. And what these individual red dots here mean are basically upward motion. So this is perpendicular to the coral surface. There's actually upward motion of some of these small particles determined by PTV. And the red ones means that these are actually downward motions as determined by PTV. And correspondingly, we can determine the oxygen concentration in these specific locations. And what comes about is that actually down here, where there's upward motion, we actually typically have high oxygen concentration. And where there's downward motion, we have low oxygen concentration. And this can be quantified by looking at the diffusive and advective flux, where, again, the advective flux at the upward motion down here is higher and lower at the site of oxygen consumption. And so I think the working hypothesis of this is now that perhaps the ciliary movement by these corals is actually coordinated, perhaps, to move oxygen from production sites in a way where the symbiogeneity density is high and oxygen concentration might even become cytotoxic to areas that are in dire need of oxygen to actually accelerate metabolism. So in a way, there might be connectivity between these complex biological architecture that we can start to uncover perhaps by use of sense-PIV. We are applying these now to other organisms. Here's a blue mussel where we seeded them with sense-PIV particles. This is really just early work here, but ultimately what we can see is similar to what was expected in a way that the exhaust plume of this blue mussel is really has a very low oxygen concentration, and the surrounding environment has a much higher oxygen concentration. So I think what we can do now is really probe in a way the use of oxygen by, say, for example, marine invertebrates in benthic environments. Okay, so I think I went a bit over time here now already. So basically, I showed you a bit about the recreation of abiotic environments, how we use temperature to probe the interplay between biological heterogeneity and an abiotic stressor, and also that we're engaging in basically making many of these other recreations of abiotic environments to test their combined and singular effects. And one thing that I'm excited about, particularly in the context of environmental toxicology, is actually supplying stressor regimes in natural scenarios or more quasi-natural scenarios. So in red here being the actual temperature profile measured in the field, and in white the chemical profile measured in the field. And by superimposing this, we might be perhaps creating multi-stressor regimes that are much, much closer to the real world. And we also want to expand the use of SensePAV not only to measure oxygen, but also temperature, pH, and other gases, as well as reactive oxygen species. We recently got awarded a grant for this, so we're hopeful that this might actually be true technically, at least in the near future. So with this, I want to thank you for your attention, the invitation, and happy to take any questions.
Thank you very much, Lars, for the great talk. So I think we can start a question part. If you have a question, you can either raise your hand or directly unmute yourself and ask.
Lars?
Hi.
Hey, this is Marco.
Hi, Marco. Hi.
I guess I'm gonna see you tomorrow. Hey, not tomorrow, next week.
Yes,
right?
pretty soon, yeah.
Okay. So great talk. The last bit when you were talking about the patches on the surface of the corals, I was wondering whether you have been able to observe for long enough to check whether perhaps there is a dynamics in terms of the high concentration regions becoming low and the low concentration regions of oxygen becoming high, or are they fixed for a long time on the surface of the coral?
Yeah, so good question. We didn't investigate this as of yet. So I think the next, kind of series of SensePAV particles that we will develop hopefully be much more homogeneous, and I think that will enable us to, for example, stimulate photosynthesis. I think that's something that we're excited about, in a way to probe very selectively under dark and light conditions, in a way the development of oxygen, perhaps the active off transport under very specific concentrations of light or radiance patterns that might not be pertinent in others. So we have not done this, but in principle, I think it's possible. One thing that we observe with SensePAV, particularly in the context of corals, that when you seed them, you have a very limited amount of time right now to actually perform the measurement, because they start to produce mucus, at least this particular species. There might be others that are better at giving us insights here, but that's one problem that we encounter. Basically, we have about I think 2 to 5 minute window to actually perform the measurement.
Okay, thanks.
More questions?
See a hand there, Jules. Hi.
Yeah, thank you. That was a great talk. Really excited to see the individual studies of the symbiodinium and also your PIV stuff. So I have a little bit of comments on each one. So, for example, it's known that the Red Sea corals are extremely resistant to thermal stress. And I was super curious to see if you were thinking about measuring those symbiodinium and comparing them to symbiodinium that came from other reefs, which have been more susceptible to stress. So I'll ask you your answer, and then I'll ask another question if you don't mind.
Yeah, I was certainly thinking about it. So, I mean, we tested, for example, there's a lot of these clades were traditionally signified as clades taxonomically of these Symbiodinaceae, and some of which are known to be more temperature resistant than others. I think we could recover that, but I think the idea would to also really ideally bring this out into the field in some form and shape, not bring it not into the field, but taking a very small sample from a coral to actually see whether the temperature sensitivity of its host matters or directly correlates to that of the extracted symbionts. So there are systems out there now which do this in some way. So CBAS, they call it. This is work by Chris Falstra, I think, who actually use this as a monitoring tool for coral reef survival under temperature stress. And yes, I would love to go to the Red Sea in a way, and of course testing, in a way, local differences or differences in symbiote composition across geographies would be exciting for sure.
Yeah, my postdoc is in a lot right now, testing our new underwater our new microscope to measure FV over FM.
Yes,
actually,
I heard about that. Mm-hmm.
Yeah, so it would be really exciting. I wouldn't view you as a competitor. I would rather view you as a collaborator.
Yeah, I would love that too.
So I think it would be fun to imagine, maybe putting a story together. We are a little bit on the edge of wondering if we're really measuring PS2. So that's an act of sort of a confession to be. We have made the magical thing. The other thing which I didn't talk about is we've developed a number of underwater optical instruments for measuring in situ PIV three-dimensionally.
Yeah.
And this was under funding of the Navy, and the idea is the typical way is with the acoustic systems that are used for flow. But I was wondering if you would be interested. I could send you some data about. I'm not really interested in selling things. We might loan you something, but there are a number of cameras that are looking at a laser illumination, which allow us to back out 3D. I think your studies were with laser sheets, if I'm not mistaken.
That's correct.
I'm not debating the veracity of that data. I think it's amazing. But if you have any interest in 3D measurements,
no,
we
absolutely.
do have some institute.
I mean, I think it would be fantastic to, and I mean, we have thought about this. I think we don't have the money basically to do it. That's the bottom line. But I think, in a way to create a perhaps somewhat deployable system for short periods of time, right, where you can actually measure PI or sense PIV in this case by seeding very, very small amounts of particles into like the optical interrogation window. So I think spot on. I mean, we would love to do something. Sure.
Yeah.
Particularly.
And we just bought this software package for some ridiculous amount of money that these guys make in the States to back out 3D PIV. So let's talk. Thank you so much for a wonderful.
Great. Thanks, Joel. Thank you.
More questions?
Yes, I have one. Thanks, Lars. Great talk. I'm actually in New Hampshire, East Coast, and I work on environmental DNA, bit far from this, but it's very interesting if you see this, like those stressor, as for micro scale, like organism to place them in an evolutionary context, like how these will impact those microorganisms through evolutionary time. And a little question linked to that is, which other parameters or stressors or selective forces can you also measure that are more specific to the micro scale? I will think about viscosity or other stuff that could be, specific to the micro scale and therefore might have an impact that could be more specific to organism at that scale.
Yeah. So, I mean, I'm mostly thinking in the analyte space, and I think there's a number of sensors out there now, particularly electrochemical and parametric sensors that allow to measure at a relatively small scale and kind of resolution, kind of the patchiness of some of these nutrient or analytes, in that particular space. So I don't know exactly what you refer to, but I mean, for me, for example, light is an exciting one. I think it's relatively simply resolved. It's relatively simply recreated, and I think that's something that I would be very much into. We are engaging in, but perhaps not at the level that I would like to as of now. To your other questions, I mean, I'm sure there's a number of kind of really abiotic, so more physical parameters such as turbulence or, as you said, viscosity. I just personally wouldn't exactly know how to measure them yet, but I'm sure maybe even someone in this audience would know, and perhaps you do. And it would be exciting to in a way engage in that. But I think I'll stick for now to the analytes, because I think this is kind of I know what they are, and I know how to measure them, and I perhaps am starting to know more about how to recreate them.
Thanks.
Lars, I have a quick question. Thanks a lot for this cool talk. It would be a naive one. I'm not an expert on colors. Why are the sources and sinks of oxygen so well organized spatially in the first place?
So my understanding is that typically at the mouth of these polyps, there's, as you saw in the picture also, there's a very typical aggregation. The density of the packing density of these symbionts is much, much higher.
On the inside, on the...
So kind of where the opening is. So, and not only that, it's also dynamic. So they can, they can actually shrink, they can bring them closer together, under different light conditions. It's a very dynamic system, right? This is not static. So, what we might see in 1 second, as being high concentration of oxygen might change after 2 minutes to something that's not, simply because of the dynamics of the system. But typically, the symbiont density is higher at these polyp inlets, is my understanding. And so that's where typically then you would expect the oxygen concentration to be higher simply due to photosynthesis.
That's fascinating. Thank you.
I have another question, if you don't mind, Marco here.
Yeah.
So, if I understand well, your Sense PIV work on excitation by UV, right? Okay, but I assume that this is near UV.
Yes,
UVA
395. Yeah,
exactly. So PS2, if I'm not mistaken, has a decent amount of absorption in that range. So. And I guess that in order to get a visible response from the particles, you need to crank up enough the UV excitation. Do you have then a problem in terms of your measurements of photosystem 2 efficiency, because you might be inducing things like photoprotection or photoinhibition?
Yeah, good question. I think I would have to go into the paper exactly what the timescales of measurement were. I think the light sheet is yeah. I don't know whether it's actually temporally separate. So I don't know if it's a very short pulse or whether it's a continuous excitation. I would have to go back to and actually check that. So in principle, yes, I think you're right, Marco. You would ultimately stimulate, PS2 activity and photosynthesis in general by the application of that light. And that's and not only that, you would probably also damage it, right? I mean, that's the bottom line. I think PS2 is very sensitive to near UV and UV in general. So I think not only would you stimulate, but you would also long-term probably perturb. And we kind of know about this a bit, for sure. So one thing that I think the sensor chemist knows and can do, you can develop sensor chemistries that are not PTT FBP based on but I think it's palladium is another central that actually does a more redshift. So what that allows you to do is to actually stimulate, for example, with other wavelengths that are not typically used in photosynthesis, such as green light. And you still pick up in a way due to the Lux-Stokes shift of this system, than infrared. It just requires a bit of rethinking in terms of, what kind of laser line do you need and all these types of things. But in principle, I think you can make a more biologically compatible sensing system that circumvents some of the problems that you mentioned, Marco.
Okay, thanks.
There's another quick, maybe naive question. What is the biodiversity of symbionts inside a typical coral? How many different species are we expecting?
Yeah, so I mean, honestly, I'm not an expert in this, and some other people really are. So I don't want to leave myself out there. My very basic understanding is that depending on location, depth, environmental profile, it's probably between 4 to 10. Okay. But again, I might be, not entirely correct here. I'm sure there's exceptions to this, but this is kind of the broad literature kind of that I kind of absorbed from this.
Sure. Thank you.
More questions? Okay, there seems to be no more questions from the audience. So I think we can thank once again Lars for the great talk.
Thank you all, and look forward to meeting many of you again soon.
And one last announcement for the next seminars. The next seminar is